up here, I said I'm David Wallace, and I'm working with my mentor Kevin Kipp in Professor Wine's lab in the Department of Molecular Cellular and Developmental Biology. So we're working on polycystic kidney disease and pretty much just trying to figure out what's going on. A lot of it is not really known, and so we're just trying to figure out what's happening and how to stop it. So as you can see here on this first slide, we have a normal kidney on the right and a diseased kidney on the left, polycystic kidney. So obviously you can see that it's characterized by all these cysts that are fluid-filled, and the kidney actually will enlarge and eventually fail. All right, so ADPKD, or autosomal dominant polycystic kidney disease, is a common genetic mutation and disorder that affects about 12.5 million people in the world. And what happens is these kidneys start filling with cysts, and they expand, and eventually they can't compensate for whatever is going wrong, and they just shut down around age 50. And so as you can see from these pictures, you have just a normal kidney, and in about 50 years it fills with these cysts, and pretty much just shuts down because it can't function the way it used to. And this, right now there's really no treatment. There's just dialysis and a kidney transplant, but the dialysis is not very, a very good life, I guess, and transplants take for a long time when you're on a waiting list. So we're trying to find some other treatment to fix it. So in order to find the cure to go from these cystic kidneys back to normal looking kidneys, we really have to figure out what's going on. We have to figure out what these mutations are doing to the kidneys, why are they filling with cysts, why is this happening, and then figure out what's the best way to stop it, what can we treat people with to get this to go back to normal. All right, so this is the main system that I'm working with. The main protein that we study is PC1. It is, the full name is polycystin 1, but it's where about 85% of the mutations in this disease occur. And so we are concentrating our efforts at looking at this one protein. It is a transmembrane protein, so it's in the membrane of the cell. And from previous studies in our lab, we find that there is this cytoplasmic tail portion of the protein that is actually cleaved off, and it goes into the nucleus and induces transcription of certain genes that promote survival. And so there's a kind of like high rate of survival associated with this disease. So cells aren't dying when they're supposed to die. They just continue to grow and get larger, and then the kidney will fail. So pretty much what we've also discovered is that once this portion of the protein comes off and goes into the nucleus, it can again be cleaved in the nucleus in normal healthy cells, and this is how the body normally regulates between cells that are supposed to die and cells that aren't supposed to die. So if it goes in, it doesn't get cleaved, then the cell lives. If it gets cleaved, it might die because it doesn't have the survival signal, so it'll be apoptosis. So that's pretty much from previous data what we have been looking at. And so my work is mainly to determine if this is actually what's happening in the kidney, if this is actually true, and just to see if we can stop this survival signal from happening. So there's mutations in this portion that comes up and goes to the nucleus that prevent it from being cleaved, because we've identified a cleavage site on it, and there's mutations all around, patient mutations all around that site that we think are preventing it from being cleaved, and that's why this survival signal is always on. So just a model of what's going on. We have PC1 in the membrane, it gets cleaved. The tail portion goes to the nucleus, and it can either, if it doesn't get cleaved, it promotes transcription of certain genes that leads to survival, or it gets cleaved, it doesn't do anything, it gets shut off, and the cell will die. So this is kind of how, too much of either one of these things is a bad thing, and so we're trying to figure out if this is a way that the body normally regulates how cells act. So this is one of the main methods of looking at how, seeing what is happening and testing if this is true. This is how a luciferase assay works. So this is if we're going to put in, we're going to get certain genes to be overexpressed in the cells. You can put DNA in, and then this is a way to observe if those genes are actually being overexpressed, and see what the result of that is. So the first step in this is to put in a plasmid which is just circular DNA that has your whatever gene you're trying to turn on or look at is in there, and attached to that gene is also a gene that promotes formation of a protein called luciferase. And whenever your gene of interest is being expressed, that protein will also be expressed. And so you can, so what you can do then is you can put on a substrate that when it interacts with luciferase, it lights up, and then you can measure that light. So whenever that light is there, your protein of interest is also there. So you can see how much is being turned on, how much is being turned off. So this is just a way of measuring what's going on. So yeah, pretty much the measured light just correlates to how much protein is being expressed in the cell. So this is my first bit of data. What we did is we put in that tail piece into the cells and to see if this STAT3 gene, which is a survival gene, is being upregulated or what's happening. So you can see that when you put in the tail this and activate this pathway, it shoots up, you get a lot of survival signal. If you put in a DNA that's not coding for anything, but it's just an empty DNA, this is just like what would happen in normal cells. So if you activate the pathway, it only goes up to here. So you can see that this tail is enhancing the signal by a lot. And so then what we did is we took the two halves of the tail, just like the first half, it's pretty much cut right in the middle, because that's where the cleavage site is. And so the first half we put in, and then we put in the second half. And you can see that the first half does pretty much nothing compared to normal cells, and the second half does much less than the full tail when it's intact. And you can see that even though this does have some sort of extra activity compared to the normal, it is much less than the full tail. So this is kind of a good indication that this may be how cells regulate this protein, because if it gets cleaved, then you can see that obviously it doesn't have nearly as much activity as if it was left intact. So that was my first little bit of data that we did. The rest of the stuff I've been doing for this project is to create another system for studying this disease. And usually when you put in DNA into cells, they express it for a short amount of time. It's called a transient transfection. It only expresses for about a couple days. And then cells, once they start dividing, they just kind of like dilute the plasmid, and it doesn't get expressed anymore. But occasionally, and very rarely, the cell will actually incorporate that plasmid into its genome, and then it will always express it even when it starts dividing. So this is a really rare event. If you have a plate of cells and you transfect them, you may only get one cell that actually takes it into its genome, even though you have 10 million cells on a plate. So it's a really, really rare event. And what you can do is, if you can locate that one cell that has the stably transfected DNA, you can select it and put it onto another plate and grow it out. And then you have a whole colony of clones that all express this protein stably, and you never have to like retransfect them. They will always express this protein. So that's kind of cool. So that's kind of what I'm trying to do now. I'm trying to put in this, this mutant where we've mutated the cleavage site on that piece of the protein, and so it can't be cleaved anymore. And so we're trying to kind of set up a system to test if this mutation is really what's affecting the kidneys. And so the cool way to select for that one cell, like how do you find that one cell that was stably transfected? On the plasmid that you put in, you also there's also a resistance gene for certain antibiotics. So if you just throw an antibiotic on there, the cell that took up the gene into its genome will have resistance to the antibiotic, and all the other cells won't. So if you leave them on the antibiotic for a couple of days, the other cells will start to die, and the one cell that had it in its genome will not die. And then you can see it on the plate under a microscope. And then once you locate it, you can use a filter paper dipped in trypsin, which is just a chemical enzyme that you can use to pretty much it breaks all the tight junctions in cells. So cells usually like to stick together, so they kind of separate. And so if you put this filter paper with the trypsin on it, they'll start to come off the plate and then detach from all the other cells, and then stick to the paper, and you can just pick the paper off and put it in a new dish. And then all your cells will be in the new dish. And so that's kind of cool. And then you just leave them on selecting antibiotics just to make sure that you only have the ones that you want. and then you can grow your colonies out. So that's pretty much what I've been doing for a couple of weeks. This takes a really long time to do. And so we're in the process now of testing these cells to make sure they're the actual cells that were stably transfected. And so I do not have that data yet, but that is what we're doing at the moment. So yeah, that was the end of pretty much what I've been doing. In the future, we can just observe if these mutants, if they are resisting apoptosis pretty much. So if they can't be cleaved, then there's always going to be the survival genes turned on. So if you try to make them apoptose, maybe they won't as much compared to just normal cells, or they'll just protect them from, maybe they'll die slower or something like that. We can test that. We can also observe the cell type that I'm using develops cysts in itself, so maybe the cysts will be bigger, or there'll be more of them in this uncleavable mutant, and we can observe that in 3D culture. And so in conclusion, we see that cleavage seems to limit the upregulation of these survival genes, but we need to confirm that this is actually what's happening, so we're going to use this system to test it. And that's pretty much it. So I want to thank my mentor, Kevin Kipp, for taking me on and dealing with all the mistakes that I make, and Professor Wimes for hosting me, and the rest of the Wimes Lab for really mentoring me and showing me how to do certain things, and I've really learned a lot from them. Also the DoD and NIH for their grants and funding, EUREKA for helping putting all this together, and EUREKA and CSI. So, thank you.
Questions? What types of cells did you use?
Oh, I'm sorry, I forgot to mention that. These, I'll have a picture of them. This is a cell type I'm using. They're MECK cells, which are made in Derby canine kidney cells. They are a Cocker Spaniel kidney cell line. We also use HEX cells, which are human embryonic kidney cells as well.
Is there, it said you put resistance to an antibiotic. I assume that there is another way of getting rid of the cells should they not be what you need to, right? Should it be cause to worry if something were to happen? Like if the petri dish would break or something like that while you work?
I mean, if the petri dish were to break and your cells would be contaminated from other things, so you wouldn't want to use them anymore. This is just like the quickest way to get what you want. It's really the easiest way, because really the only thing is that should be left on the plate is what you want, and then you can grow it up. But yeah, I don't. Do you have anything like specific that you were? Okay.
I think the question more is, isn't using antibiotics for this purpose dangerous because it might cause things you don't want having resistance to them to build resistance to them? Is that what you're trying to get at?
Like what kind of things?
I don't know.
The cells that we use, really the only thing that are in there are the cells that we're dealing with. There's no like bacteria or viruses in it, because that could be some variable. Everything's done in a hood in a cell culture room that's pretty kept clean, so that you don't have contamination, because that could be some other weird variable that could play into the system. So you try to eliminate that completely. Yeah.
Could you go back to maybe your first opening slide? I have kind of a two-part question. So,
This slide or?
the one, no, just like your title slide.
Oh, okay.
So, at what stage, so would the goal be to take a fully diseased kidney back to a normal kidney, or would the overall goal be to determine how to stop a kidney from becoming diseased?
At this point, actually, either would be beneficial, but really the goal is actually to be able to revert it back to normal. We've actually seen success in mice models for certain things that have been cystic. So like in this picture here, we've actually been able to treat it with certain things to get it to actually more resemble this, which is kind of cool, because the cyst will actually decrease in size. And that would definitely be more beneficial than just preventing in the first place. But yeah, I mean, at this point, either would work.
And then the other, I guess, kind of along that line, and that's pretty amazing, is what stage do people get diagnosed for this disease? And so do people oftentimes only get diagnosed after it's full-blown cyst, or can you detect it instead of at 50 years, at 25 years, that you were gonna develop
Yeah,
this?
it is an autism dominant disease. You can be tested, there's like genetic markers that you can test for to see if you are going to develop the disease. If you somehow don't know that you know, if your parents have it, you're very likely to have it, so you can be tested for it. If you don't know that you're going to have it, you can, sometimes the first symptom is actually back pain because these kidneys are huge and they just, you'll go in and say, my back is hurting, and you realize that your kidneys are 4 times the size they should be. So, I mean, it could happen, you can be tested, but if you don't know, so sometimes this is the type of disease where it's not like your life is sort of like degrading slowly. You are kind of fine until about 50, and then it just shuts off. It's kind of like, it's stable, and then all of a sudden it drops. So it's, by the time it's dropping, it's almost too late. You have to do dialysis right away or kidney transplant. So, yeah, it's kind of tough.
You said you've been spending weeks trying to create like the new stable cell line. Have you had like some? Is it like you go from no success to immediate success, or are you having slow success? I'm curious about that.
It's just a really slow process because you have to transfect them, and then the antibiotic that we're using is actually it kills them about a week, and you have to leave them on there for a while because some cells will be transiently transfected, and so they'll have resistance for a little while. And then once they start dividing, and they stop expressing the gene, then they won't have resistance anymore. So you have to wait for those cells to die as well. And so it kind of just takes a while, and then you have to deal with these weird things where you have to like dilute them onto different plates. And so because you only want to get really one cell, because you want a clone of just one cell. And so you don't want to have, you don't want to accidentally pick 2 cells. So you have to really ensure that there's, very few cells on every plate, so you have a very good chance of just getting one cell. So it just takes it's just a long process, and yeah, I should know by the end of the week if we did it right, but we'll see.
So, thank you,